Today we embark on the epic quest and process known as DNA replic. Wrong video. Known as DNA replication. This is a process in mitosis during the S phase in which DNA is just duplicated or replicated, which is quite obvious from the name. So we start with DNA as it's found normally or naturally in the nucleus, which is coiled and double helixed, but then we're going to branch off into two separate strands. And let's designate this as the 3', this is the 5', which means following these strands, this would be the 3', and this would be then the 5' end. So in order for these strands to be separated, it's done with an enzyme called helicase. And they're separated because each strand is going to be replicated individually. So it's separated by this protein called helicase at a spot called the replication fork. But in being separated, it's actually causing a lot more strain on the coils and the double helixes down the road of the DNA molecule. So in order to alleviate that strain, we have a protein called teloisomerase. And again, all this does is it just alleviates the strain caused by the helicase, which separates the DNA strands. But what keeps them from bonding back together? Once they're separated, why don't they just come back together and bond and ruin the whole process? Well, that is prevented by a bunch of single-strand binding proteins, which bind to each strand, actually, of this new DNA, or the parental DNA. It binds to each of these, and all it does is just stops them from coming back together and bonding again. So let's look at first, in a replication, let's look at what's called the leading strand. And this is the strand that ends in the 3', and we'll differentiate more between this leading strand and down here the lagging strand. And you'll see quite clearly why each one is called which one. But let's just look at the leading strand first because it's a lot more basic and it sets down the principle of what's going to happen. So you start with something called RNA primase, which lays down a primer. So we'll call this RNA primase. Getting a little out of room here. Which lays down this primer right here. And it actually lays down RNA nucleotides. And this is going to be a problem that is addressed later. Well, not necessarily a problem, but it's just something that is addressed later. They're going to be switched out for the DNA, but that's to come a little later. And then we're going to add on with something called DNA polymerase 3. We're going to add on DNA nucleotides complementary to the strand that already exists. And if we look at the strand, we're going to see that if this is 3', this means that this one is 5', and it means that this one is 3'. And this makes sense structurally for how DNA is, because you can only add on nucleotides to the 3' end. So it's quite easy to see how the leading strand is simply using DNA polymerase 3 and the primases, but the DNA polymerase 3 specifically to add on the nucleotides of DNA in the direction of the replication fork. And it's really quite simple because that's kind of it. But we get a problem because the lagging strand ends in the 5', well, not necessarily ends, but it kind of, for the purpose of this diagram, it ends in the 5' end. And so when you're trying to put on new DNA, how do you do that? Because it has to go that way, because the new strand would end 3' over here, so you have to go that way. What happens is you put on another RNA primase down on this strand, which then of course puts down the primer, and then again you use the DNA polymerase 3 to add on the nucleotides as you go in the 3' direction, which is all correct, 5' to 3' direction, adding on only to the 3' end because that's the only thing that's possible. But what you do is you do this at different spots. You do this a bunch of different times because now this doesn't have anything, so you have to add another RNA over here, RNA primer or primase and primer over here, and another fragment going in the 3' direction. And you do this a bunch of different times. As this is separating and as this is moving along, you're continuously putting down new primases and sending up new fragments. And these fragments are actually called something. They're called Okazaki fragments, named after the Japanese scientist who discovered them. And that is also a fundamental difference in the lagging strand versus the leading strand, is that they lay down Okazaki fragments, while these just continuously lay down nucleotides in the direction of replication. So again, lagging strand is Okazaki fragments. It is, as we can see, discontinuous, and it is also, as we see, away from the replication fork. The replication happens going in the opposite direction as the replication fork. So now we have a little problem, as I said earlier, is that these are RNA primers, or they are RNA nucleotides because of the RNA primers. So in order for them to be switched out with DNA, is you have another enzyme. So let's say we have some more primers, the Okazaki fragment, another primer, some Okazaki fragments, and let's look at this primer. This primer is RNA nucleotides. So we're going to act on it with something called DNA polymerase 1, not to be confused with DNA polymerase 3, which adds on nucleotides to the 3' end. This is DNA polymerase 1, which switches out the RNA nucleotides and puts in DNA nucleotides. And we are almost done at this point. Because the only problem is now we have, let's say right here, technically DNA polymerase comes first, but another enzyme called DNA ligase now finishes up the job. All that needs to be done now is that these DNA nucleotides that were just switched in for the RNA nucleotides, which were switched out, aren't quite bonded to the rest of the DNA nucleotides. And we need to keep this continuous. It's not much of a problem up here, although we will put in DNA polymerase 1 and the DNA ligase for the primase, or for the primer that is up here. But there's only one of them, so it's really not that big of a deal. You just put in one, you bond it with DNA ligase, and you're good. This one, you bond it with DNA ligase and make it continuous, but you have to keep doing that for every single primer that comes up. But that's basically it, because at this point, now you have this leading strand, which is continuously moving in the direction of replication, and it's pretty much done. You have one primer, and then this continuous strand. On this one, on the lagging strand, is it lags behind a little bit, because the 3' is that direction. So you have to set down multiple primers, and they lay down Okazaki fragments, and in this way it's discontinuous. And it's kind of lagging behind, and the direction of replication is away from the replication fork. And then again, you use DNA polymerase 1 and DNA ligase to take out those to take out the RNA nucleotides, put in DNA ones, and then kind of finish up the job by further binding them together, by binding those sugar and phosphate groups back together. And that, in a nutshell, is basically DNA replication. It's just a series of a bunch of different proteins and enzymes that act to just separate the strands, put down these primers, put down the nucleotides, and on the lagging strands, a bit different, it's just in fragments, but then that's it. You have the complementary strands read in the 5' to 3' direction, and your DNA is pretty much replicated.